How are you? Good? Good, okay. I'm going to talk today, very different kind of talk, but with, I think, a bunch of overlap. I'm very excited to be speaking to you about my favorite topic of how to extract knowledge from big data, in this case, the big data of genomics. I was attracted to this field because it's really exciting. It's had all the wonders that I found in my computer science career, and I said, you know, I really would like to do something beyond the iPod and iPhone and Mac OS X. Although wonderful things that those are, though that is in fact an accomplishment, it wasn't the future, right? And the future, in some sense, is how do we take this enormous capabilities that were just emerging about 5 years ago, and I realized, wow, we're going to see an incredible explosion of data. How do we interpret that into meaningful knowledge? And so I'm going to answer about 5 questions, right? What is the problem, and why should you care, right? And I'm not going to talk about all the biomedical problems, but I'll talk about an important one about cancer. What do computer scientists, or what need to know about biology? What should you know as computer scientists? And then I'm going to talk to the people who aren't here, mostly. What do biomedical scientists need to know about data and big data and computers? And then how do we transform that data, that genomic data, into knowledge that is actually applicable and can be used to treat people? And so that notion of a medical intervention is still extremely at issue here. But of course, there's many uses beyond that, and we'll talk a little bit about that too. And I'll also conclude with a few remarks relevant to people who make this fundamental platform software. So what is the problem, and why should you care? Okay, very clearly. Cancer is really the second biggest cause of death. Heart disease is number 1. Cancer is number 2. Stroke, which is often part of heart disease because of problems with your cardiovascular system, really is connected, and it's broken out separately because it's the way it's experienced. And then there are a bunch of other problems. You know, if you don't die from bungee jumping, one of these things is likely to get you. So heart disease, as important as it is, and it has an important genomic component, right? It is not primarily a big data problem. It is a big data problem, but you'll see that the texture of the data is enough different to tease it apart from cancer. And so in terms of actual numbers of how many people die, right, it's about 570,000 people a year. More than 500,000 people die every year from cancer. 1.5 million people get it. So, you know, only about a third. About a third. And that's a serious number that die from it and whatnot. We want to see these numbers go down. And we haven't been able to knock them down too much. And part of that has been because of the complexity. And I'm going to explain a bit about the complexity of what's going on so you have a good sense of this. But this is what's driving that enormous healthcare cost increase. It's one of the huge increases. This and Diet Coke, Coke, regular Coke. You know, it's that diabetes and such, something we need to do something about, but you know, this and problems. So what are the interlocking problems here? Well, as a patient, right, you have to make choices. You have to make choices based on information that your coach, your doctor, your family members, the press and whatnot, that give you data, and to make those choices between chemotherapy, surgery, radiation therapy, when things are in the early stage, or into experimental methods, clinical trials, alternative therapies. These are very difficult choices, and the data to do this is actually not all that readily available. For a few cancers, it's very obvious. For the vast majority, it's not. And the doctors really actually don't have an enormous amount of databases to pull from. Part of that is because the medical system is broken, but part of that also is that the complexity of knowing who you are as a patient is, in fact, a very complicated situation, so that the normal factors that would help you drive a decision, for example, what do you want to eat tonight? What did you eat last night? What information that you readily are available from Yelp or Google or other online resources cannot be used in the same characteristic to help you decide which is the best treatment for me, which is the best therapy, which drugs have the most likely chance of success. And even when it's made available, of course, patients don't necessarily know how to interpret it. Doctors don't know how to interpret it. Probability is actually a very complicated issue that most people have not received professional training in. I actually think that probability should be taught in elementary school, not in graduate school first. And so it's really essential, I think, to bring some of these in. But that's not this topic of my talk. That's the topic of another talk. OK, so I hope you believe now that it's extremely important that we understand some of these things, because we're going to learn something about cancer biology now. OK, something you all know, I hope, which is the central dogma of biology. And that is simply that DNA is translated into RNA, and RNA is then translated into protein. The first part of the process is called transcription. That transcription process is an information process. And the RNA process is an assembly process into a three-dimensional protein. Proteins, of course, are the elements in your body that make up both the structure, and they are actually themselves information processing components. So if you think about it, right, proteins are, in fact, part of a network of systems that exist in every cell and between cells for part of this cell communication and logic processing. And they have all kinds of incredible shapes. None of that is particularly important for this topic of this conversation, but it in itself is a beautiful and lovely field. We represent, of course, information using molecules. These molecules, adenine, guanine, thymine, cytosine, are simply assembled into the lattice, and that lattice, of course, is extremely stable. It consists of these phosphates and with a sugar backbone. And that one reason why DNA is so prevalent in this is it's extremely stable, right? Even in the face of multiple types of radiation, it is capable of representing information over a vast period of time. We're now able to sequence the DNA from Denisova individuals who lived 60,000 years ago, and we actually have a very excellent assembly of what their genomes were. Not complete by any means. There is, of course, some degradation, but it's such a robust molecule that it holds information for 60,000 years. Try to store your PDF file on your little thumb drive for that long, right? Good luck. So what does that mean to cancer? OK, so in cancer, we have this. What happens is that there's a progression of degradation that happens typically with one of a number of genes that suppress the tumor. And every cancer has this progressive process where it begins and it gathers steam as more and more mutations accumulate. That process of accumulating mutations is occurring all the time. There are, of course, error-correcting proteins in your body. There is redundancy. And the ultimate thing is the body actually, immune system, comes along and sees, this is growing out of proportion. Let's zap it and kills the cells. When that fails, and that can fail for any of several good reasons, then, of course, you have cancer. And one of the things is that the tumor itself acquires a novelty where it's able to tell the immune system, oh, ignore me. I'm not the problem. I'm a wound. I'm healing. Well, that process is one that means that, of course, it's best for us to catch cancer very early on. Cancers caught in the very earliest stages are easily treated. Cancers caught in the latest stages are much more difficult to treat and typically fatal. I mentioned that oftentimes these are caused by single mutations that occur. So here we're looking at melanoma. This individual on the left has melanoma of their body. And one of the interesting things about this is it's caused by a single point mutation in the BRAF gene, a tumor suppressor gene, in the V600. We actually had an off by one error. It's at 599 position, but we still call it the 600 position. And it's like debugging, right? So we found this one location. We found a drug that treated this particular protein, and bam, 15 weeks later, a targeted therapy managed to treat the patient. Wow, isn't that amazing? Well, unfortunately, we didn't get all the cells in the cancer. What happens is that some of the other cells came back, which didn't require this particular mutation. And so at 23 weeks, the patient is back again with new tumors. The drug is no longer doing any good, and the patient succumbs to it. So while we had some encouragement that we're making progress, this BRAF inhibitor drug is extremely useful. In fact, it's not necessarily a war we always win, okay? And that is part of the big issue we need to help with. Okay, so what does that mean? Well, one thing is that because the mutation process is ongoing, the cell is acquiring all of these mechanisms of drug resistance. These actually make the cells more fit. They are supercells, right, the cancer cells. They are more fit than the regular cells of your body. They, in some sense, have decided to no longer go by the covenant of sacrificing their own health for the benefit of the body that they're in. They said, no, I'm going to grow beyond. And what they do is they acquire these additional capabilities, reducing the amount of drug they uptake, improving the damage repair. They can actually improve their own damage that the drugs may induce on DNA. And it can actually eject the drug out of the cells faster than we can get to it. What, in fact, is going on here is evolution. And it's these mutations that begin to accumulate over time, initially that are benign mutations that don't do any harm, but occasionally you get a driver mutation depicted in orange here. And those driver mutations help improve the fitness of the cell. So we talked about evolution before. Here's an example of where it actually is a predictive model, where we can say that the improvement in fitness of the cell increases its ability to grow to larger and larger, and for it to actually remove some of its capabilities to fight, the body's capabilities to fight the cancer. That fitness curve means that there's kind of this process of a divergence of a population. And just like you might have multiple populations, multiple species in soil or in a forest of a community of any kind, you actually have this process where, which is the light, there's the light, where we go from a few initiating cancers, and then additional driver mutations occur, and the population increases to different populations, so that by the time we're able to detect the cancer, we're not treating one disease, we're treating many diseases. How many we're treating, that's an argument of some kind, because we really don't necessarily have yet a terminology to talk about this. Like the bacteria, we may have many independent species in some sense of cancer. And so each of these driver mutations may actually represent a different disease. We may have one treatment that kills off one population of the cells, but then the other ones now are able and more fit to go and move on. We can see this sometimes even under a microscope. We start off with a nice 46 pairs of chromosomes, 46 chromosomes, and what we get later on is some bizarre number of very misshapen chromosomes. So we actually can see that under a microscope, but we can certainly see that molecularly. Okay. What does this mean? Where is the action? And I want to tell you about pathways. So we talked a lot about genes. A gene codes for a protein. But proteins interact in pathways, in networks. And as network engineers, as I think all of you are, and as computer scientists, as many of you are, I think you'll appreciate that what we're looking at here is a highly adaptive, redundant system. That redundancy is fundamental to the way evolution works. It's fundamental to the crosstalk and the communication, and its ability to accept novelty. Just as you might have redundant code in a program, right? Code that's mostly being used or is only used occasionally, right? Evolution is a cut, copy, paste engineer. It's a cut, copy, paste engineer. It makes whole copies, makes minor modifications to some of the existing code, and it runs along. And that modification, that slow process, actually is a positive thing as a species level, but not so good necessarily in fighting cancer. So what we see, in fact, is that cancers are caused not by single mutations in one gene in one place, but by multiple genes in a pathway. So, in fact, I don't know if you can see these numbers. So, for example, we'll see mutations for, in the P53 pathway, the one that's the most. We'll see that, in fact, that sometimes the mutations are 35% in TP53, 7% in the MDM gene, 14% up in the amplification. But overall, we see it in 87%. This is glioblastoma data from the Cancer Genome Atlas. And that's a huge number. That's like a huge number. So we shouldn't necessarily think of it as being something wrong in the p53 gene, it's in the p53 pathway in the circuit. And so we actually see this along in 78% of the cases, and in, go back one, and in the RAS pathway. And so what we're talking about here is, in fact, multiple redundant mutations in a given individual, right? Add up these numbers, obviously greater than 100%, right? Multiple redundant driver mutations, and we need to treat each of them. So there's a lot of complexity here. So the goal here is to build a database with all these variations, with all of this into a massive database from the patients that doctors and researchers can use to interpret. And we are doing that. This is a good thing. We are doing that. This is a very big part of a project called the Cancer Genome Atlas. And this is where big data comes to medicine. So this is, the outreach of our world into the biomedical world. And so what the Cancer Genome Atlas is, just as you would have an atlas that describes the overall structure of a map, maps, I think are big today, we are trying to, we build this structure to come together and bring about an enormous amount of data and many sequencing centers all working together, all driven by this better than Moore's law capabilities of genomics. And that's a really exciting thing, because if you can turn your problem into into a sequencing problem, you're going to get the benefit of this. So a big part of this is a center we're building at UC Santa Cruz called the Cancer Genome Hub. There's a poster on this done, and I won't go into too much details. Our partners, Anai Systems, built a wonderful infrastructure that allows us to download massive amounts of data in a reliable way using a BitTorrent-like protocol. And the big part about this that you need to understand really is that the data is there. So what are we going to do about high-throughput genomic technologies? Well, in essence, there's so many important aspects of the problem that have to do with how the proteins and the chemistry interact. But if we can turn them into this DNA sequencing problem, then we're able to. I'm going to skip ahead, because I don't think you need to know too much of this. But I think, and Titus spoke about how we do the shotgun sequencing. The interesting thing about this is that there is this wonderful notion here where what we're doing is a lot of string comparison. And so a big part of the processing is to use that string comparison, not just in terms of coming up with a single assembly, but in fact, we also turn this information into knowledge. That knowledge allows us to do things, for example, to find the gaps or the duplications. If you have a duplication of a gene, because of the cancer, because of the mutations that happen, because the actual copying process itself has made additional copies of the protein, then you essentially have something we call a dose-response gene. In other words, the more that we have to, gives us insight into the fact that this greater dose number of copies of the gene actually means that we have to increase the medication or find some other strategy to treat the patient. We build this into pathways. We built a terrific set of pathway databases. We have about 30,000 nodes in this pathway database these days, acquired from public sources. But realize that 30,000 node pathway database is still only a fraction of the potential million nodes we estimate is actually in the graph. So we only have, if you will, the interstate system of the graph. We don't actually have the minute locations of all the streets and towns. So what does the data deluge look like? We have all these interactions. We have to put these together into some kind of semantic meaning where we take the exome sequences, the methylation data, the structural variation data. And so we have this problem of the complexity of how these interactions occur. And of course, as you know as computer scientists, that with 2 to the n combinations of n data types, that becomes very difficult for our poor biologists friends to cope with. We need tools to help them manage it all. The way we've chosen to do this, going back again, is to the fundamental central dogma of biology. We take a simple idea. For example, here MDM2 downregulates TP53 gene. And that begins in the dogma model. And we can now plug into this all these data types. Because if it doesn't follow the central dogma, it's not biology. Right? That's one of the interesting things. There are other parts of loops to the biology graph that we didn't talk about, but it's still there. And so what we're able to do is to build this massive model, we call it paradigm, which allows for this gene model integration to allow for integration of copy number, expression, protein level, protein activity level to make these graphs. These graphs are each node of that 30,000 node graph consists of this structure. And what we do is we do this integrated pathway analysis. And what we're able to do is to infer by turning everything into a probability, layering it over the graph, and then doing a technique called expectation maximization, where we are inferring the other missing nodes that we cannot measure in the graph. That expectation maximization process allows us to sample, if you will, a portion of the graph structure that our machines are very capable of doing and infer the regulatory and other control state. And in fact, to conduct in silico experiments, once again, as Titus said, that will save the biologists many years, decades of time. So the analytical strategy here is very simple. We're going to turn this massive amount of data into these signatures, and we're going to draw inferences from similar and distinct patterns, where we cluster signatures to find the patterns and such. Now, the problem, of course, with this is we have to prevent bias, and we have to prevent multiple test false discovery. And this is a huge issue with statistics and a terrific conference by itself, but I'll give you a little flavor of it. I am a believer in very basic statistics done at massive scale. There are many people who do fabulous approaches with machine learning technologies, support vector machines. I think all of these are great. I don't feel we've exhausted the t-test yet, right? The t-test, still damn good, you know, still good for another 100,000 miles. And the t-test applied to vast numbers of genes in huge order and doing simple hierarchical clustering, I think is a big boom for us. So we take this information, we take this dichotomization, where we turn things into two classes, we do this classification, and we turn it into heat maps. Biologists can deal with numbers if they're presented as heat maps. Okay? So that's a big, you know, lesson learned. You know, the math, little scary to the biologist, very simple stuff, you know, done in aggregate, where we simply do Euclidean clustering over this data. You know, lower your, if you're servicing biologists, right, don't have to go very hard into the math textbook to impress. What does the heat maps do? Here's a publication we had in Nature, which allowed us to take the mutations and the behavior and the effectiveness of aromatase inhibitors. Aromatase inhibitors essentially block estrogen in breast cancer. Very important therapy, huge issue. We had not had a good model for when it works and when it didn't work. Right? And what this paper did, in constant work with UC Santa Cruz did with Washington University, essentially was able to take the map of pathways, most of which we knew and understood, fill in a few extra details of genes that we had not known were involved, and to say, if the mutation occurs in this part of the pathway, the drugs fail. If the mutation occurs in that part of the pathway, it succeeds. If your mutation, and you can be tested now for these sets of mutations, you know, don't bother with aromatase inhibitors, move on to a completely different class of drugs, and good luck. With the top portion, you know, aromatase inhibitors are worth a good shot, okay? And so that's a huge issue, saving suffering and money. All right. I talked for a very brief time about multiple test hypothesis. What do we use in biology? We don't have good closed-form analytic models, right? What we do instead is we do permutation testing. And so what that means is we're generating false models, and we know they're false. We've done just simply random variation, and we're looking to see how similar is the answer to the one that we got from the true data. And then we look to compare that. And that process is one where we typically do, for example, 1,000-fold false permutations, and then compare that whether or not it's in the top 0.05% of the drawer. What's interesting about this is lesson for the computational people, is that most of what I do is false, right? It's mostly done on bogus data to simulate. So that, I think, is a huge difference, perhaps, from the physical world, where you're not doing this type of bootstrap permutation model. So a good deal of the computation. Can we use that to make our computations more efficient? I think that's one of an open question to look at. So how do we transform this knowledge into medical interventions? Right? It's not enough to find these discoveries. So I believe in something in the power of social networking. I think social networking is an enormous new tool that's allowing us to break down the barriers between conventional sources. Carol talked about the silos between patients, doctors, academic research, and we need to do this. And I think social networking can be a big part of this. System I'm working on is called Medbook. It's essentially, the idea is to create this kind of an apps platform where creating these applications takes advantage of this genomic data and brings it into some degree of a actionable cause. And the idea, of course, is to have both a way for patients to be involved with this, for their doctors, because doctors ultimately need to be involved in every aspect of the medical care process, for academic researchers and clinical researchers to work together to understand, translate this data in the vocabulary they're used to of heat maps and other forms, and for academic developers, non-biologists, to be able to develop these algorithms over this biological data and to be able to run it and test it in an efficient environment. So I think this is a huge opportunity for us to take what we know about big data and turn this into actual pragmatic treatments. Okay, so I'm going to wrap up very quickly, but just to say that I think there's a kind of a machine architecture here that you need to know about. And that is that really there are, in medical care, there needs to be 3 zones of data. I think of them as the red zone, the blue zone, and the green zone. Without going into great detail, the red zone is the HIPAA, patient identity protected zone. The blue zone is the genomic data, where the data can live de-identified, but still may be individually identifying. And the green zone is what we think as a conventional internet. And those 3 zones need to have this full circle process where data is gathered from the patients, is managed by the doctor and healthcare system inside of a red envelope, as it were. All this, of course, is nothing more than VPN and good two-factor authentication. The blue zone is genomic information, can be done in mass, but it's de-identified, so it allows for non-HIPAA medical researchers to access. And then the green zone is open to the public, or at least not necessarily having the close, tight collaboration that the blue and the red zones have. So I think this is an interesting architectural principle that I'd like to propose. So in conclusion, what we're trying to do is make personalized medicine a reality by bringing social networking. Patients will gain visibility. Doctors will actually be able to translate what they genomic information into cures. Clinical researchers will get larger cohorts, statistically relevant ones, and app developers can get a market for their development. Couple quick remarks for the XLDB community specifically. So what do? We do everything with Unix command line tools, right? And we're compute bound because of the null permutations, not really because of the amount of data. Databases are typically stored as CSV files, very trivial kinds of data flow. Computations, my favorite tools happen to be 1,000-node compute clusters. I have terabyte machines available. I thought I would use them a lot. I haven't had to yet. Maybe as the data grows, as some of these better algorithms come into being, I will use them more. Conventional languages, R is a big one. We talk about a language not being useful to us until it's R complete, meaning it has the full library of R algorithms and functions. And we do a fair mix of open source and proprietary code. Metadata and version management is a nightmare, huge nightmare. We do a very poor job of it, and we're not taking advantage of the read-only data in the environment. There's no use of previously done large operations. We typically do it over and over again. And that's because we have no real good way to store the provenance of the computation. Okay, so what are requirements for you guys to work on? Well, for one thing, this is both an online transaction processing system and an online analytical processing system. So it's got both merits to it. We can use, obviously, a combination of technologies, but we need the data flows to flow between. The bulk of the data, you can think of it as a hypercube. Sometimes we're looking across cancers. Sometimes we're looking at an individual patient. Sometimes we're looking at very large cohorts. Sometimes we're looking at slicing and dicing. So all that is, of course, the kind of language that the big data world is used to using. But we key it by a multigraph, by this graph of pathways and such, to actually extract out what we think is relevant and interesting. We, of course, do want the ability to slice and dice across many dimensions. A lot of the code I tend to write is simply reorganize the data yet again into a more efficient form and then process it through. That's a huge bulk of my time, I think. The workflows and conclusions must be traceable. Traceability is something Unix tools stink at, and we really need help with that. And I think part of this is to be able to share workflows, part of a huge part of where we're headed. That needs to be closely tied to the publishing and reputation models. And with that, I will give up the control and answer your questions later.
Thank you.